Well, it's that time. We gotta talk about stuff. So, let's see, announcements. Lab, I'll be in lab tomorrow if you have questions about the final lab report, so do stop by. Also, I think it's time for everyone to wash out their fly bottles. So clean up those bottles this week or next week, I don't care. So I can get them all sterilized and ready for next year. So lab, we're just kind of cruising through right now. We have a couple things due coming up. So Friday is the exam, which you've all looked at now, right? It's
10 questions.
10 questions, 10 points each.
Easy.
Oh yeah, okay. If you say so, I'll look at yours with an especially critical eye. These are essay questions, so one thing I want to mention is it's fine if you get together with your buddies and do the research and think about it, but I don't want to get bored reading them. So don't just write one essay and everybody copies it, okay? Write it independently, each one of you. I want to see your words in the explanation. So do that. Let's see, then the plan for next week. Let's see, so you're going to turn in this exam on Friday. That means I'm going to spend all day Saturday grading it. Oh boy. And then you're going to turn in the final lab report Monday. And I will be grading that frantically as well. So that on Wednesday, yes, on Wednesday, I will be able to give you all your tentative final grade. So the plan is for next week, Monday, I'm going to wrap up a few things. I'm going to discuss a few matters. So yeah, we're not done lecturing at you yet. And then Wednesday is wide open. We can discuss your grades. We can discuss just about anything. I won't be recording on Wednesday. So yeah, you can say whatever you want without it going up into the world. And that goes for the people who don't normally show up. Yeah, you guys. If you don't normally show up, that's okay. But I'm just saying, Wednesday you might want to stop by, and if you have questions about anything, that'll be your last chance to hit me up with that. Okay. Is there anything else I need to tell you? Any questions? We're going to finish this course. It's going to happen. We're getting there. All right, so today what I want to do is talk about Chapter 24 and cancer genetics. Yes, because I love to end the class on a really uplifting topic. But cancer is kind of an important phenomenon, and genetics is at the heart of it. Now, I'm going to give you just a rough overview, a survey of a couple of genetics-related things in cancer. You know, we do have a cancer biology course here. That's taught by, by Rachel Johnson, or I think Danny Boland is doing, yeah, Danny Boland is, I've taught it before, but they're better experts on cancer, so yeah, it's a good idea to take it from them. So I'm just trying to set up that course here. So what are we going to talk about? Well, we got to first say a few things about normal cell development. So this is just a diagram of an early embryo. And the key thing here is that it is normal and healthy for cells to proliferate and grow, especially at specific times in development. So what we see here is here's a blastocyst. It's just a bunch of indiscriminate cells sitting in there. It's got to go through a number of divisions. This looks like what, 16 cell stage or thereabouts? You've all got a few more than 16 cells. So you know we got to divide like mad to produce the individual that you are now. So we got lots of cell division, but also what's going on here is that cell division has to be regulated and controlled. It's not just a mass dividing madly, because you can have to form things like this. This is a blastodisc. This is, by the way, humans, what they look like. So this is where the embryo proper is going to form. And you got to build these extra embryonic membranes and sacs and all this other complicated stuff. We are also going to go through things like gastrulation, that's illustrated here, where cells are going to delaminate from this outer epithelium. This is the epithelium here. They're going to delaminate to form mesoderm, which is going to contribute to things like muscle and bone and blood. And then we also have another layer down here. So we're a sandwich, two epithelia wrapped around a mesodermal mass. All this has to be precisely coordinated. And again, we want lots and lots of growth in these embryos. That's the only way it can work. Got to divide like crazy. So what we're going to do is build these tissues. I'm just going to give you a very brief overview of the histology here. That what we have in these organisms is we got layers, and the layers are organized in very precise ways. This, for example, is an epithelium. Look how neat and tidy it is. Got all these cells just stacked up, adherent to one another, with an absorptive end and a luminal end, etc. This would be, for instance, the lining of your gut. You got this kind of tissue there. Now, epithelia are clearly important. You got epithelia on your skin, right? That's a squamous epithelium, but it's got a columnar base underneath it. So you need these epithelia, and these epithelia also have the property that they have to be constantly growing. The epithelia have a rough life. The ones on the outside, you're constantly shedding, right? I bet you the room here is full of dander from people shedding bits of skin. You are constantly shedding the intestinal lining. So yeah, when you go poop, that's a significant part of that is you. It's bits and pieces of your intestinal lining just being lost, also bacteria from things like that. So these have to proliferate constantly. This makes epithelia kind of a danger spot for cancer. Cancer is where you have cells dividing uncontrolled. And here we got cells that have the first requirement, they're dividing rapidly. And if they lose control, yeah, you get all kinds of nasty cancers from that. Another important component is connective tissue. So connective tissue is all that matted stuff. It's things like tendons, it's things like bone and blood, all that's connective tissue. And there it's a little more complex. So connective tissue has a significant acellular component. It's like you see over here. We got these collagen fibers all over the place. So you don't have to worry about collagen getting cancer. It's not dividing, it's not a cell. But then there are these cells in there like macrophages, parts of the immune system. There must be some, yeah, we got macrophages. We must have fibroblasts somewhere in this illustration. I don't know, somewhere. Anyway, fibroblasts are also there. They're the ones laying down all that extracellular matrix. Those have the potential to become cancerous. If you've got an active cell that's dividing, that's a danger. A necessary danger. You need this stuff going on. But yeah, we've got to worry a little bit about that. So we got connective tissue. There are connective tissue cancers. There are specialized connective tissues. Things like cartilage and bone. Yeah, there's cartilage cancers. They're rare. And there's bone cancer. You've probably all heard of that. There are, of course, blood cancers like leukemias. that exist. So these are all the cellular components do divide and have the opportunity to go cancerous on you. I mean, that's a terrible way to put it. The division is good. You want your cells to, you want to be growing, you want to be replacing damaged or lost tissues, all that's great stuff. It just opens an opportunity for errors to creep in. Okay, we also have muscles. Muscle cancers are relatively rare. Can you guess why? They do occur, they're just not very common. Again, okay. So, what do you know about muscles? What is the property of muscles? We told you epithelia are dividing and replicating all the time. What about muscles?
It's hard to grow muscles.
Yes, muscles don't grow. They tend not to divide. If you're thinking about, oh, you're an athlete, you get out there and you pump iron and all this stuff, and you get big muscles, your cells aren't dividing there. They're increasing the number of fibrils inside the individual fibers, but you're not getting a lot of cell division. So you're virtually no cell division. So you're in pretty good shape here. Where we think most of the danger comes from in muscle for cancer is that there are also stem cells. And your textbook mentions that one of the common theories for the origin of cancers is the stem cell theory, that it's arising from stem cells that have gone bad. And so those are a candidate for generating muscle cancers. Then we have neurons. You look in your brain, you look in your peripheral nervous system, and you look in there and you find all these lovely neurons here. Are neurons a great candidate for going cancerous? No, why not?
They don't divide.
Yeah, cancer cells, or neural cells, are done dividing, so they're not going to. Yeah, at your age, they've just started dying. Sorry.
Oh, good.
Yeah. Hey, how do you think I feel? You've got a surplus, though, so don't panic yet.
Yeah.
So, neuronal cancers are really uncommon. However, your brain also can, and also your peripheral nervous system, contain these things, supporting cells, glia. I used to work with a person who studied glia, and she got really upset if you called them supporting cells, because they have so many rules. They have a lot of roles to play inside the organism. So, yeah, look at all the things they do. And glial cells do divide. So you can get things like gliomas, which is a form of cancer derived from glial cells. Okay, so there's different degrees of susceptibility to cancer in different tissues, and often it's directly associated with their rate of cell division. Okay, let's start with a horror show. What this is two micrographs. On the top is a normal breast. So what you have in a normal breast is you have ducts. Obviously, it's a glandular tissue, and those ducts and those milk-producing cells are embedded in a stroma matrix, which is connective tissue. So that would all be fat and fibrous connective tissue holding it all together. That's what it's supposed to look like. In breast cancers, you get this kind of situation. And you might be saying, where are the ducts? Where are the nice tidy tubes that are supposed to be running through the breast cancer? And no, they're disorganized. So in this case, the epithelia, typically it's the epithelia of the breast, have gone cancerous, they've gone amok, they've started dividing and spreading, and they're no longer organized in the nice tidy you want for a glandular tissue. So that's a problem. Yeah, this is where you get lumps. Breast cancer is often easily treated with surgery if you catch it early enough. So yeah, you should be doing those regular breast inspections You find a tiny little lump, and it's a tiny little lump, still has millions and millions of cancer cells in it. And if you remove it before it spreads, you're probably okay. Although then you also have to worry about genetic predispositions to cancer. We'll talk a little bit about that in a moment. Okay, here's another case. So this is a section through the skin. So there's that squamous epithelium I mentioned, that's the outer layer. And then down here in the dermis, we've got connective tissue, and we've also got, for instance, things like melanocytes right there. So those are the cells that this summer you're probably hoping to get out and get some sun, and they'll expand and they'll grow. And they'll darken your skin to a nice golden bronze, right? Unless you're my kind of Minnesotan where you avoid the light. But yeah, this is responsible for all that kind of behavior. Because these cells are very responsive to environmental signals and are capable of dividing rapidly, they are also a danger. This is melanoma. Yes, go out and get the golden brown skin, but avoid this. If you see this kind of thing, you're in trouble. Melanoma is a particularly dangerous kind of cancer because it spreads rapidly, it proliferates strongly, it's prone to metastasis, which we'll get to in a moment, and it can do horrible things to you. I had a sister-in-law who died of melanoma, and it was a long, slow, painful process where they started off by lopping off her arm to try and contain it, but it spread to the rest of her body before they did that, and yeah, it was fatal. So watch yourself. Check your skin out when you're out there sunning too. Sunburns seem to increase the likelihood that you'll get melanoma. So tan sensibly, all that kind of good stuff. Okay, so melanoma, that's not good. Oh, look at that horrible picture. This is a mouse lung. So we're not looking at human stuff here. So this is a normal mouse lung. This is a mouse lung that contains all kinds of these little black granules. Those are all melanoma. So we think of melanoma as a cancer of the skin, right? But it can spread in the process called metastasis to other tissues as well. And that's what's particularly deadly about it. It can spread into your blood system and your organs. And look at this poor mouse. It's got melanoma and it's spread to their lungs. And as you might guess, that's not a very functional lung. Especially not since the mouse has been killed and it's cut out of it. Okay, so those are things to watch out for. You got excessive growth, like we saw in that breast tissue, where cells have become disorganized and overgrown. And then we have metastasis, where the cancer cells not only grow like mad, but then they erupt out of the affected tissue and start spreading throughout the body. And that's where it becomes extremely difficult to control. You can lop off a piece of skin that's got melanoma in it, which is terrible, it's awful, but it saves your life. But once it's spread to your lungs and your kidneys and your liver, no, you cannot lop those off. You're stuck with those. So then we're stuck with things like radiation and cancer or chemotherapy to try and clean up the problem. Okay, so what do we think is going on in these cancers? This is a diagram from a book called Cancer, or Biology of Cancer, which is from a massive paper that summarizes everything we know about cancer. And it's a diagram illustrating what's going on. What is a tumor like? A tumor is a collection of really sick cells. Tumors are disgusting. I've seen tumors excised, and I've opened them up myself with a scalpel, and they're just full of rotting dead cells. There's blood and goo and pus and all kinds of things in them. So tumors are really ugly when you get up close and look at them. What we see illustrated here, though, is kind of a cartoonish representation of a tumor. And what you find inside the tumor are cancer cells. These are cells that are typically genetically damaged in some way and are dividing rapidly. But what you also find are healthy cells. So here's an endothelial cell, for instance. So these are little capillaries and blood vessels that pass through there. And the capillaries and blood vessels themselves are not cancerous. But one of the things that cancer cells may do is secrete angiogenic factors and induce the growth of blood cells into the tumor. So that's what you're seeing there. You also find things like connective tissue cells. One of the things the body does to try and constrain a tumor is it will pack it full of connective tissue. They just kind of wrap it up in a sheath. And that's all part of the effort of, hey, we got to put brakes on this. We got to confine this. This is also common with fairly benign things like cysts. Yeah, you get a bunch of responses to that. Your body will do what it can to confine it. The problem with cancers is they keep growing and they can break out of the bounds of your nice little connective tissue sheet. The other thing you'll find in there is immune cells. Your immune system does not like cancers. Behave yourself. Okay, so your immune system does not like tumors. And so you will have loads and loads of inflammatory cells all packed in there, doing their best to combat the tumor. So a tumor is very much a hodgepodge of different kinds of cells and cell types. It also contains stem cells. So cells that can divide and perpetuate the tumor. So we've got all this going on in here. And typically this is going to be it's going to be inserted into some kind of matrix. Like if it's in your skin, it's going to be surrounded with connective tissue out here as well. So we're going to have all this material packaging it up. And unfortunately, because the cells are dividing rapidly and want to spread and get out of that system, you can get invasive tumors in that region. So that's what we're seeing here. Here's our tumor. Breakout cells have escaped and are spreading the cancerous cells into the immediate neighborhood. So you got a tumor in the breast. It leads to more tumors in the breast. So not a good thing here. And finally, what can happen is some of these invasive cells can break so free. They can get into the lymphatic system, they can get into the circulatory system, and they can spread to remote areas and invade those and start new tumors in those regions. Yes, is this a really optimistic sort of talk so far? No, it's not. We have all kinds of treatments for these things now, and year by year the cancer survival rates get better and better incrementally. Yes?
So what would be the difference between like this kind of tumor and a benign tumor? Are the cancer cells just not present?
Oh, in a benign tumor, well, do we even call them, we don't necessarily call them cancer cells if it's a benign tumor. Okay. It's kind of precancerous.
Okay.
So what you've got is a bunch of cells that are dividing but may not have the genetic prerequisites to become truly dangerous to the tissue.
Okay.
And the whole idea there is, yeah, you got a benign tumor, we can either leave it alone, and unless it grows so big it causes problems in that region, we don't need to cut it out. But if we cut it out, we're done. It's gone. Mm-hmm. A malignant tumor has got more active growth properties, and you want to get it out of there, but you still have to worry afterwards that maybe some of those cancer cells have escaped and are spreading all over the place. So yeah, Yeah, so there are tumors, for instance, that are not cancers. Last time this worked so nicely, but today. Anyway, there are lots of cases of tumors that are not cancerous. My grandmother, for instance, had a tumor, and they diagnosed it as benign, and they just go ahead and hack it out, and she was fine. So, yeah, there's a range of effects here. Okay, so this is what we worry about. We don't want things to spread all over the place. Okay, for genetics, what do we care about? Well, there's a couple of terms we need to know. These cancers all have a genetic basis. Some cancers are familial, that is, you can inherit a predisposition to acquire them. You may have heard of this in, for instance, breast cancers. There are certain breast cancers where if you've got a certain gene configuration, you're more likely to have a subsequent error that will lead to full-blown breast cancer. So you've just got a predisposition. This also fits with the multi-stage model of how cancers work. It's not just, boom, one mutation, you've got cancer. No, it's combination mutations that build up to lead to cancer. So we got some terms here. First one is some genes are called proto-oncogenes. What are these? These are genes that typically are involved in promoting cell growth and proliferation in healthy, normal tissue. This is what you want. These are good genes. Nice gene. It's doing important things for you. So you've got lots of proto-oncogenes that are sitting there, for instance, telling cells that, oh, you've got an injury, start growing, expand this layer of cells to fill in this injury, etc. So we like those. And that's basically all proto-oncogene is a gene that says grow. But the important thing is controlled. It's got a set of controls that regulate when it gets turned on. An oncogene, you do not want an oncogene, okay? You want proto-oncogenes, but you don't want oncogenes. And what's going to happen is that if there's a mutation or damage that modifies a proto-oncogene to strip off those controls, then you've got a cell that's going to induce uncontrolled cell growth, that is cancer. And we don't want that. In addition, there's another class of genes called tumor suppressors. And these are genes that in the normal healthy tissue limits cell growth. These are the brakes in your cells. So even in a healthy tissue where you may induce proliferation and growth, you've also got a set of genes that say, no, stop, you've reached the limit. They also do things like control the ability of cells to grow into other tissues. So for example, there are tumor suppressors that control growth of cervical cells so that they grow in a particular specific region. And one of the things a gynecologist will look for is, do you have cells that are growing into abnormal tissues? That means your tumor suppressor genes in that cell line are not working particularly well. So lots of predispositions we can look for. Okay, how do we get oncogenes? Just take it for granted. Oncogenes you do not want. You want proto-oncogenes, but it's fairly easy to convert a proto-oncogene into an oncogene. And there's three main mechanisms listed here. So one is just gene amplification. So we have lots and lots of proto-oncogenes. If you get more of them, that can bias the cell towards growth. So you can get a strong response just by amplifying the number of proto-oncogenes you have. Another is regulatory mutations. So the important thing with these proto-oncogenes is they're supposed to be controlled, right? You're supposed to turn them on at certain times in certain places and then turn them off once their job is done. And so if you have a mutation, the regulation of those genes, that can convert them to oncogenes. And in these cases, you can actually pull out the gene, the coding region of the gene. You can look at the protein that's being produced. It's a perfectly normal, healthy protein. It doesn't contain any mutations, but it's being misexpressed. And that's what leads to cancers. And then finally, you can also have coding mutations. So a mutation within the gene proper, within the coding region of the gene, that produces a defective gene product. Okay, let's talk about gene amplification first. Oh, look, molecular biology. I'm sorry if any of you are suffering through that now. It might bring back flashbacks or something of molecular biology. But okay, what we're seeing here in this fairly complicated diagram is we are looking at a human gene, HER2/neu, which is a proto-oncogene that can be converted to an oncogene. And we're looking at it in a couple of different ways. We're looking at DNA with a southern blot, RNA with a northern blot, and protein with immunoprecipitation. And each of these is a different patient. Okay, so we're looking at different individuals here. The key thing to take away from this is that you've got a marker for HER2/neu, and you stain in various ways for it, and you can see all kinds of variations. So here, for instance, this patient, look, it's got a lot of DNA here. There's been some kind of amplification of that gene. Here we got normal levels of the DNA. But here we got somebody with a normal level of the DNA, but look here, the RNA has gone crazy. So something is misregulating the expression of the gene, even if the gene itself is fallen. And these are protein immunoprecipitations. And again, you can see there's differences in the production of the proteins that we can also see in this immunohistochemistry down here. So it's not quite as simple as just, oh, let's just go look at the gene and see how many copies are there. Because it can vary in its regulation at different stages. So this is just to let you know, it's a messy business. All right, in this case, what we're doing now, we're looking at that same gene, we're looking at HER2/neu, and we're looking in human breast cancers. This is a really depressing graph. I'm sorry. Because what you're seeing here is you're getting patients who come in who've been diagnosed with breast cancer. So here we are at the beginning of their treatment, and we examine their levels of the HER2/neu. And what we see here is a case where you don't have amplification. You just got the normal number of HER2/neu genes. And this is what's depressing about it is, yeah, I'm sorry, over the course of a few years, you get more and more death. This is just patients dying off. We're looking at survival in this graph, and it's just dropping off until you reach kind of a lower plateau. This is where the doctor will go to the patient and say, yeah, we think we've gotten rid of the cancer. We've cleared it out. The chemotherapy has worked. The surgery has all worked. And some minority, less than 50% of the patients are doing okay after a couple of years. See what I mean by depressing? But what's even more depressing in this case here, the patient comes in with breast cancer, just like the ones with the blue line, and we look at their genes and we discover, this patient has an amplification of HER2/neu. They have greater than 5 copies of that gene in their system. And look what happens there. This is just a tragedy, a huge loss of life. Very rapidly, within a year, to get down to this lower plateau. Again, the doctor would go to the patient and say, good news, we've got it under control. You're probably going to do okay. Come in every couple of years and get tested again, right? That's what's going on here. But before that, you get this massive loss of life very quickly. Yeah, so having an amplified HER2 new gene is not a good idea. Not much you can do about it, but it's one of the things doctors will look at. Here's another case. In this case, we're looking at pediatric neuroblastoma. So several years ago, I got into a long conversation with a pediatric oncologist. And what I said to her was, at first was, this has got to be the most depressing job in the world because you're treating kids with cancer and some of them are going to die. And it's, I couldn't do that job. And she said, no, she was very optimistic because every year it gets better and better. Treatments get better and better. And they're doing a better and better job of helping kids make it through. And kids are so plastic and robust that you see a few kids die, and that's tragic. And you see other kids that recover and go on with no signs of cancer. They grow into adulthood. That's got to be a really optimistic sort of thing to see. So she told me, no, it's not as bad as you think. The other thing is that every year, hospitals do incredible numbers of statistics. They are measuring everything. And every year they see those statistics improving. Not dramatically, but just this gradual incremental improvement. And so that's also very encouraging. But here what we see is a pediatric neuroblastoma. These are cancers that arrive in the periphery. They're typically involved with, for instance, Schwann cells, the cells that wrap around neurons. They happen peripherally. And they're not good to get. But here, look at the blue line again. Yeah, you've got a pretty good survival rate here. We're talking 90% of the kids are treated and they survive. They make it to adulthood. So that's good. Where they get these successes, though, are in kids who don't have huge numbers of copies of this one oncogene, this proto-oncogene, and MYC. If the kid comes in and they assay his tissues, they find, oh, you got more than 10 copies of NMYC, you notice this does not look so good. Again, another rapid drop-off, reaching a plateau. And 10% survive in this case. So like I said, it's a scary job, but it does have it has the bright spots up here, and it's got the sad ones down here. Okay. So amplification affects this stuff. These are people who are overloaded with his proto-oncogene, and it leads to these cancers. And there's lots of these. And this is a table of all these different genes that are found to be amplified and associated with these cancers. You can see that some of these, like look here, CAB1, ERB2, GRB7, that's 3 different genes. But they're linked. They're actually close together on a chromosome. And what has happened is a little chunk of the chromosome has been amplified to make extra copies of it. And that's where you get the problem. So these are all tied together. And you don't know which one is specifically associated with, for instance, gastric cancers. But you know it's one of those 3. Maybe it's the combination of all 3. So you see lots of linkage listed down here. Okay. And you also see here, this is the human chromosomal location, so we've got that all worked out. But the amplification is not good. You don't want amplification of oncogenes. Okay, what about gene mutations? We also get variations like that where we mutate genes and they cause problems. Here's a simple example. This is the RAS proto-oncogene. And if you look at the sequence of RAS, there's all those amino acids and all that. In the healthy cell, it's got a glycine right here at this position. And if you get a mutation that converts that glycine to a valine, look at that, it's a single nucleotide transformation. This becomes a full-blown oncogene. It's got some other variations in here, but don't worry about those. So yeah, we can go from a proto-oncogene to an oncogene with just a single nucleotide change. There's more of these RAS oncogenes. So these are looking at human tumor cells again. So this is another common routine thing. If you've got a cancer and they're doing surgery to remove a tumor, or they're doing a biopsy to pull out a bunch of cells from it, they can now screen for various genes. That's for, do you have a common mutation in a particular gene here? And RAS is one of the ones that's implicated in a lot of cancers. So as you see here, this is a proportion of tumors carrying a point mutated RAS gene. So look at that. You've got a patient with pancreatic cancer. It's 90% chance it's got a mutation in RAS. At the same time, you got a patient who's got kidney cancer. Not so likely. Still 10% carry mutations in the RAS gene. So there's a huge amount of variation here. This is one of the problems with treating cancer, is that there's more than one way to get a cancer, right? So here we got a patient, and we're saying, okay, it's 90% likely you've got a mutated RAS gene. They look in his tumor, and no, he's one of the 10%. He doesn't have a mutation in RAS, but he's got cancer. So obviously he's got a mutation in something else. So it takes some sleuthing to figure out what's going on here. But you can see again, there are these percentages associated here that sort of suggest, yeah, here's what's going on. This is a contributor to a certain degree to these kinds of cancers, but it's a little bit variable. Okay, what about regulatory mutations? So those coding mutations, it's kind of obvious. You got a proto-oncogene, you change the proto-oncogene sequence, it disables some of its control functions, you get an oncogene. We also have regulatory changes. Here's a really well-known one. This is kind of interesting. This is a particular kind of cancer called Burkitt's lymphoma. It's a really unpleasant cancer. It's most common in children. What it does is it causes massive overgrowth and swelling of the lymphatic tissues. So kids with Burkitt's lymphoma may have grossly distorted faces because they get these massive tumors growing in the lymphatic ducts. It's less common in adults. When you look at the incidence of Burkitt's lymphoma, you can see it's really common in equatorial Africa. And this is kind of cute in a hideous, horrible way. This is where you have malaria and the incidence of all kinds of malarial diseases. Furthermore, when they study these patients here, they've also got Epstein-Barr virus. And so we think what's going on to induce these cancers is you've been infected with a virus transmitted by mosquitoes. So it's not malaria, it's Epstein-Barr virus you're getting. And that is inducing mutations, changes in regulatory genes, and thereby inducing cancers. When you look more closely, oh, this is kind of interesting. What you discover is that these patients have a high incidence of translocations. You remember, we talked about translocations. So here's normal chromosomes, here's chromosome 8, here's chromosome 14. Chromosome 8 has the MYC gene, this proto-oncogene. And over here on chromosome 14, there's actually a couple of other chromosomes that have these molecules as well. You have immunoglobulin H, the heavy chain of immunoglobulin. In your lymphatic tissues, you all know this, right? The lymphatic tissues contain a lot of immune system responses. And so in these tissues, they typically have cranked up IgH. They want to make lots of heavy chain immunoglobulin. So you've got this gene turned on, very active. You got this one, which is supposed to be tightly controlled and not so crazily active. And then you get this translocation that you see over here. We're going to swap out this terminal end of chromosome 14 and stick it on 8. And we're going to take this end of chromosome 8 and we're going to stick it on 14. And you get MYC right next to the IgH molecule. And so it adopts the regulation of IgH. And you get constitutively active MYC gene products. So yeah, again, why would it be in the lymph glands? Because that's where you've got IgH turned on usually. If you get this in your skin cells, no, they're not making IgH, so it does nothing. But once it gets into the lymphatic tissues, then you get overactivity of MYC. It becomes an oncogene. Okay, so this is just illustrating the same thing, that you've got your normal chromosome 14, it's got the heavy chain immunoglobulin sitting there. You got your normal chromosome 8, which has got a tightly regulated MYC gene sitting there. You get a reciprocal translocation. Now, MYC is sitting right next to IgH. Okay, you've also passed along a piece of this to chromosome 8. So you get all kinds of problems. Furthermore, once this happens, you will often get further mutations that affect the expression of MYC. Because when a cell is dividing, there's more opportunity for it to acquire mutations. So you've got this hyperactive cell that's acquired this unregulated MYC gene, and it will accumulate more mutations that can fall in this region, and you can get worse and worse conditioning of the MYC gene. Okay, lots of translocations have been identified that affect this. So here you can see some examples. This is MYC. There are these others. Let's see, I'm going to give you another example in a moment. But these other molecules, these oncogenes, are activated by a translocation that produces these kinds of problems. So leukemias and lymphomas are really common in these cases. Again, these are things that are affecting blood cells. Now, I told you about this one before. Y'all remember? Philadelphia chromosome, classic example of this. So this is, again, a reciprocal translocation between chromosomes 9 and 22. And what it does is it puts the proto-oncogene c-Abl. Okay, where's c-Abl here? There's c-Abl, little red dots they've got. And you put that on another chromosome where it is put under control of the BCR gene. And then you get constitutive activity of this proto-oncogene that now becomes an oncogene, and you're going to get cancers. This one, again, was identified in Philadelphia, the Philadelphia Children's Hospital, where they noticed a heck of a lot of their children with leukemia, when examined more closely, had this peculiar translocation. Okay, what this does, this is just illustrated here, you got this Abl tyrosine kinase. This is our proto-oncogene. We got this BCR, which is constitutively active. It's always active. And what this is illustrating is there are multiple places where the insertion can occur. Not every kid in the Children's Hospital of Philadelphia had exactly the same mutation. They had translocations, but the breakpoint in BCR could occur at various places and produce various permutations of that hybrid BCR-Abl protein. Yes?
Did they find any correlation between where the mutation occurred to if it was survivable or more lethal?
Why, yes. Did you look at my next slide?
No.
You're just psychic. That's good. What they found is Yeah, this uses a few too many colors to make it really clear. But what we see here is still C-able, and here's C-able over here on this end. And here's BCR, this bit over here. We'll get variable bits of this attached on here. And what you find is that different leukemias are associated with different treatments, with different abilities to treat. I can't remember which one is the most dangerous. But yeah, you got acute lymphocytic, you got chronic, that should be chronic myelogenous, and chronic neurotrophic. And they are produced by different fusion proteins in this case, and they also have different survivabilities. Okay, so yeah, this translocation business can produce all kinds of different combinations of oncogenes for you with variable effects. All right, again, there's lots of them. Here's the one we just talked about, BCR-able, right there. So it can produce these different kinds of leukemias, but there are also all these other fusion proteins. And again, this is one of the powers of genetics and molecular biology, is that they can study these things. You come in with some terrible leukemia, and yeah, the scientists on staff will get all excited and take samples of your genome and try and figure out exactly what has been changed to produce this condition. And then, of course, the regular doctors are sitting there trying to fix you. So that's the good part. All right. Another thing we find is viruses that do this stuff. So viruses, let's see here, for instance. Oh, here's one you've probably heard of, human papillomavirus, which was in the news because we've got a vaccine against that, which is a good thing. Human papillomavirus, what it does is that if you're infected with this, it can insert itself into the genome of your cervical cells and other cells, but it's in the cervical cells that it causes the problem. And it can damage other genes in that genome and lead to the production of an oncogene. And then cells start dividing, and you've got cervical cancer, which we'd all like to avoid. Okay, you can see that there's others that did. We mentioned Burkitt's lymphoma. Lymphoma is one of those. Non-Hodgkin's lymphoma is also another well-known one. Kaposi's sarcoma, which you may have heard of as common in, for instance, HIV infections. Same thing. You got a virus. It's invading your cells. It's inserting itself into your DNA. And given an opportunity, it can insert into endothelial cells and produce these opportunistic cancer infections. Yeah, Kaposi's sarcoma is really, really rare, except in cases where you've got some kind of viral disease promoting its production. Okay, that's enough depression. Let me just wrap it up here. So again, what I'm telling you is that cancers are complicated. There's oh, come back. Wow. This is what happens when you push the wrong button. Okay, cancers are complicated, and there's lots of different tissue types in there. That there are cancer cells, but inside of a tumor there are also healthy normal cells, which tends to complicate the treatment a little bit. For example, I mentioned angiogenesis, that tumors are secreting hormones to trigger the growth of blood vessels into the tissue, which means that often excising a tumor is a very bloody business because it's full of blood vessels, healthy, normal, hard-working blood vessels that are transporting hemoglobin through the tissue, and that's not good. There a number of years ago, one of the hot new ideas for treating cancer was anti-angiogenic drugs. If you treat somebody with a compound that prevents angiogenesis, it doesn't affect you normally. It does greatly affect wound healing, but it also puts a brake on the growth of tumors. And people were very excited about this for a while. But as I told you, there are many different ways to become a cancer cell. And cancer cells eventually figured out ways around those particular drugs. So it's got short-term utility, but lots of problems with using it. Okay, we can finish up a little early. Go have something sweet for lunch. That'll cheer you up after this. And like I said, Thursday, I'll be in the lab if you have questions about this fourth exam or about your lab report. And everything will be done by this weekend, except for the grading. I will get all the grading done by Tuesday and come into class on Wednesday, a broken man, but we'll cope. And wash out those fly bottles at some point.